And now we'll be able to move on to our next session. And I've got a great group of panelists to help us in this next session. This is our first pipeline session, where we're going to talk about treatments that are in development for FA. Can we go on to the slides? Thanks. All right, so quickly, I'm just going to introduce who our panelists are. And what we're going to do, I'm going to give an overview of kind of how we think about developing treatments for FA and the pipeline in general. And then I'm going to give an overview on each program, kind of where things are at, and then ask our panelists some questions to further clarify what's going on in their programs and how it relates to everyone in our community. And then we'll open it up to all of you so that you can ask your questions. So we have Wendy Agnese from Biogen. Wendy is the head of the U.S. Medical Rare Disease Team. We have John Cherry from PTC Therapeutics. John is the global project lead for Vatiquinone. We have Shana McCormick, who you just met, from the Children's Hospital of Philadelphia. And we have Gene Kelly from Stealth BioTherapeutics. He's the executive director of medical affairs. So thank you all so much for being here today with us. So Dr. Lynch went over this briefly. When we think about therapies, we try and target therapies from multiple different perspectives. So when we think about what's causing FA, we're trying to target therapies that address all these different issues, whether we're targeting the gene, and later pipeline session 2 is going to talk about ways that we might think about targeting frataxin protein deficiency, as well as the underlying genetic cause of FA. This panel session, we're going to focus more on the downstream consequences of frataxin deficiency, so that impaired mitochondrial function and what's going on in the mitochondria and the cell, that we can, ways in which we can kind of reverse or stop the downstream consequences of frataxin deficiency, and then evaluates how that impacts the symptoms that people with FA experience. And so this is just another way to think about that. For me, I always think of FA as a cascade, right, of things that happen. And there are 2 ways to think about it. One, starting at the gene, but you can also think about it from starting with the symptoms. What bothers you the most? And how do we help treat FA when we think about the symptoms? And that was also part of what you heard in the last session. Even though we might not have the cure yet, there are ways to treat the symptoms of FA. We can treat diabetes in FA. And we want to then also treat what's happening in the cell and what's happening at the genetic level. I hope all of you are familiar with this image. And if you're new and you're not, this is our drug development pipeline. And so this is not all the research that's going on in FA. These are programs that are in clinical development, in clinical trials. If we put everything up here that was currently in earlier stages of treatment, we'd have to have you here for the whole week and to go through all of them. And this would be a much bigger, more complicated picture, which is exciting. But, you know, we really want to focus on the things where the programs have progressed to the point of clinic. We have a first approved treatment in Omaveloxolone, and you see that at the top. And the goal here is to advance therapies from the left to the right in terms of stage of development, and go from those early preclinical stages through clinical trials to approved treatments. And the different colors as you go down the slide are different target areas for those therapies. So up at the top in this session, we're going to focus on improving mitochondrial function and some of the downstream consequences of frataxin deficiency. And then some of the other therapies that we're going to cover in the afternoon are on the latter portion of the slide, targeting the protein and genetic therapies. The website that I mentioned earlier has a much more detailed version of the treatment pipeline. I see Barbara is here. Barbara Tate's our scientific officer. She keeps that pipeline up to date. Every time there's a development on any of these programs, we put it on the website in the section for each of these programs. And so try and, have that in your kind of tabs of frequently visited sites to just go back to and see what's new in terms of development for all these programs. It'll be there. So I'm going to start with Omaveloxolone. So I hope all of you know that Omaveloxolone is a first approved treatment for FA in individuals 16 and older. But what is OMAV? If you're new to the community, what is this? How is it working? OMAV is a small molecule that increases antioxidant activity, improves mitochondrial function by activating a pathway called NRF2. And so in the context of FA, what we've learned through some of the basic science studies a long time ago is that when frataxin's deficient and there's mitochondrial dysfunction and oxidative stress, NRF2 is a pathway that your cells should be activating to help compensate for that. And paradoxically, it's not. And so with OMAV, we can activate that NRF2 pathway to reverse some of those downstream consequences of the frataxin deficiency. And so OMAV helps to restore NRF2 activity. And we saw that in cells and animal models and now tested that in clinical trials. And the clinical trials that were first done were in individuals with FA 16 and older. Those trials demonstrated safety and efficacy that got the drug approved. Biogen has helped provide us an update for today. These updates were provided for FARA to be able to present and discuss with you. And so we have some disclaimers. But again, this is an exciting time, right? We have approved treatments. I have to give disclaimers. But one of the first questions we get is, what about if I'm not 16 and older? And so we've been working on putting together plans for a pediatric clinical trial. And that's what's going to be required to expand access of OMAV to younger populations. And so earlier this year in July, we announced the opening of a phase 1 pharmacokinetic or PK clinical trial. And the goal of that study is to identify the appropriate dose of OMAV in children with FA. They project to enroll 20 individuals ages 2 to 15. It's a single site study at the Children's Hospital of Philadelphia. And the first cohort of individuals aged 12 to 15 were enrolled and have completed the study. A few weeks ago, we announced open enrollment for 2 more cohorts of individuals 6 to 11 and another 12 to 15 age group. So Wendy, my first question for you, or maybe you can help us. Why do we need a PK study if the drug is already approved? Can you explain to us why we need this study and what Biogen needs to learn from it?
Absolutely. Thank you, Jen. And it's wonderful to be here again with you all. So thank you for your participation in this meeting. So with any investigational drug, you're going to work it through different stages. And you just use the word cascade. I love that. It's a cascade of different research that needs to happen before you can get to that regulatory filing. So when you think about a pharmacokinetic study, what does that mean? What are we looking to achieve with this phase 1 program? So pharmacokinetic, when you think about that, what we're really looking to understand is if you give a drug, an investigational drug in this case, because we're studying it in a population we don't have current approval for, we're really trying to understand how the body is affecting the drug. So when you think about drug development, often we think about the flip side of that. What is the drug doing to the body? Here, we're really trying to characterize when you give a certain dose to a certain patient population, in this case, a pediatric patient population, what are those different pharmacokinetic things mean? And what we're describing there is you give a drug, how is it absorbed throughout your body? Then how is it distributed to the different tissues in your body? Is it getting to where it needs to be? How is how is your body reacting? How are you metabolizing and breaking down that drug? And then how do you excrete that? I think the vitamin D conversation, we talked about some of the ways that you can excrete different metabolites through your system. So that's really the focus of a one dose type of PK study. And it's that characterization, all those data points together that we can pull to really understand, are we getting the right dose for this very specific patient population, in this case, pediatrics? So it's a one dose. That's why if you participate in these studies, you often get a lot of labs drawn. It's because all this data needs to be collected. And then that gives us the confidence that we can move forward into a bigger study, often called a phase 3 program, to study it more broadly. So Jen alluded to the different cohorts that we currently have. So we look at different groupings of ages. So we have the 12 to 15-year-old, which is where we started. That first cohort or grouping of patients in the phase 1 study is complete. Those analyses are ongoing. And now the second 2 cohorts, we're calling it A2 and B. Those are active at David Lynch's site. So thank you to everyone participating and helping move that study at CHOP. And then the last cohort would then be our youngest patients. So that would be the 2 to 5-year-olds in the patient. And that would be the next step that will be informed by these earlier cohorts in the phase 1 program.
Wendy, is there any chance that there might need to be additional cohorts in those 6 to 11 or 12 to 15 after like this next round of testing?
So right now we're aiming for the 20 patients, as you alluded to, and that very thoughtful approach of breaking out the different age groups to really understand there might be differences in the potential doses that you see in those. We don't have that data yet. So that's where you do one cohort and it forms what you need to do in the next. So FARA announced the 2 cohorts that are active now, and that 4th cohort would then be that final cohort in the PK study. And then we're hopeful that will give us the information we need to really select that dose to carry forward into the larger pediatric program.
Thank you. And so we need to figure out how the drug's metabolized in children, if it's different than adults, and know what the right dose is going to be in children, because we still need to do what's called an efficacy study. So like a phase 3 study where the drug's given to children for a longer period of time to further establish safety, but then also to know how the drug is helping those individuals. Do we see the same response that we saw in previous clinical trials? Is that correct?
Yep. So it's a very good point to distinguish the phase 1 from the potential phase 3. The phase 1, again, very focused on what we want to learn around the pharmacokinetics, and we'll get some early signals on the safety there as well. It's important to note that those patients will receive the 1 dose of drug, whatever dose that's identified for, and then monitored over the 15-day period for the different pharmacokinetic measures. Those patients will have the option to enroll in an open label extension, but even with that open label extension, that won't give us the robustness and the data that we need to feel confident that we've established that safety efficacy profile or the risk benefit profile that we would need to bring forward to regulatory authorities. So that's where the follow-up phase 3 program would come, which would be dependent on the learnings from the phase 1. And I also think it's important to say that a program like that, Biogen or industry in general would never do that in a silo. So that's where we're really working and having continued conversations with the community, with the experts in the space, like Dr. Lynch, different regulatory authorities, to make sure that as we bring a broader program forward, that it will meet all the requirements. Because again, our goal here is to bring access to the broader patient population.
Thanks, Wendy. Another question we frequently get from the community and the global community is, how and when will OMAV be available globally? And I know now that there's been an approval earlier this year in the European Union, and the drug is now available in 12 markets outside the US. I believe all of those. I believe all of those are Europe. Is that correct?
So it's a process for sure. The teams are working with urgency to bring this forward more in global markets. That's really the goal, again, to help as many patients as we can. So US was the first approval. Earlier this year in February, we did have the authorization from the EU Union, the European Union, that is the umbrella deciding organization over 27 European countries. So it's important to note that that was a huge milestone when we think about access at the global level, but that doesn't mean immediate access in every region. So the EU authority took the recommendations. If you hear the words like EMA or CHAMP, those are the parts of the EU organization that review all the data and make that recommendation. Then the EU Union took that and approved it or gave the authorization for access. So now Biogen continues to work at the regional level globally to really work through those reimbursement and access issues that can really vary in the different countries. So that's an ongoing process that we continue to support at a global level, at the regional level. With that, that's the EU approval. We've also recently submitted filings in Australia, Brazil, and Argentina as well. So the team is very active in continuing those conversations more broadly, again, to make sure that we can help as many patients as possible.
Great. Thank you. I know it's important to continue to understand how OMAV works over a longer period of time and often what we call in the real world. And so Biogen is planning additional studies of OMAV, even in individuals 16 and older. And so one of those studies is what we call a post-approval safety study. The acronym for this study is PASS. But the idea here is when folks are coming into the clinic for natural history study visits, if you are newly taking or going to soon take Omaveloxolone, we'll be able to prospectively collect data when people start taking OMAV for a long period of time, for 5 years, to collect that ongoing long-term safety data. Wendy, can you tell us a little bit more about, kind of the importance of these studies and any additional information you want to share?
Absolutely. So in the opening session, you spoke to the Unify and the importance of that global opportunity now within the clinical consortium. So this will be an opportunity to work with those sites to, again, collect data in an observational way when patients choose with their physician to start Skyclarys Omaveloxolone therapy. And the importance there is, again, real world data. It's so rich and important to understand and make sure that what we saw in the clinical trials is translating in real world. And that's where the post-marketing commitments like a past study, the post-approval safety study, will be very important. So the goal here is going to collect longitudinal data over the course of 5 years in all of the 300 patients that would be enrolled in this study. And the key piece here too is that we'll collect data points along the way. So if patients see within the past study, the protocol, their physician at the beginning at baseline, and then months 1, 2, 3, 6, 12, and then annually after that, that gives us a really robust data collection to be able to analyze along the way and share back with the community what those learnings are from a safety perspective.
And this is a 5-year study, you said. Do we wait the full 5+ years to know what happened, or is there the opportunity to kind of learn along the way what this shows us? Because 5 years is a long
We'll take the latter approach on that one. Yeah, we'll definitely learn along the way and then share those key points back, not just with the community, but with regulatory authorities, with the medical community. I think there'll be rich learnings within that we'd be eager to share.
Great, thank you. One final.
Yeah.
Oh, okay. Yep.
Okay.
All right. You guys all heard that, right? All right, good. Um, so Wendy, my last question for you is, if someone is thinking about taking OMAV or they're taking OMAV and they're now coming up on their renewals, like if they're having challenges, either accessing OMAV or, need more information about it, are there services available through Biogen or, where do people go for help?
Happy to answer that one. So, and hopefully you can hear me online if I'm leading in a little bit more and we're not getting an echo in the room. So what we have is the REACH program. So this is our Biogen Access Program resource that supports all patients on Skyclarys. So this is something key to point out. The beginning pieces to that is if the decision is made between the healthcare provider and a patient or their caregiver to start treatment, the physician then sends a start form, which includes a prescription, to the REACH team. That's step one. We also need the signature from the patient to make sure that you want to be part of this REACH program. Those two steps activate our team within the REACH team to then support you in the entire journey from the beginning of starting treatment throughout your journey on Skyclarys. So they're there as a point of contact to go to for you. There's lead case managers, and then we have family access managers. We have them here today. So I encourage you to reach out if you have further questions than what we're touching on here. But they're really there to help you remove any barriers or questions that you have regarded to access, insurance verification. So again, point person, but the important piece of that would be the first piece is have that discussion with your physician and it's decided is an appropriate treatment for you. That's when the start form goes in, the signature is started, and then that activates our team to proactively reach out and be there to support.
Great. Thank you, Wendy. All right, I'm going to move on now. I'm going to talk a little bit about Vatiquinone and ask John some questions. But if you have questions, again, for any of the panelists, we're going to make sure we leave time. So what is Vatiquinone? Vatiquinone, you may have heard of it previously as PTC 743. We've done previous clinical trials with Vatiquinone. It is an inhibitor of 15-lipoxygenase. There won't be a quiz on that later. You don't have to remember that, but I think you'll see an evolving theme here. Frataxin deficiency, right, causes mitochondrial dysfunction that leads to oxidative stress. When there's chronic oxidative stress, which we have in case of a chronic condition like FA, that oxidative stress tries to initially activate things like 15-lipoxygenase to come in and help dampen that oxidative stress. But in chronic condition, you kind of get these vicious cycles that develop. And so 15-lipoxygenase, when it is overly activated, starts to become harmful. And so Vatiquinone inhibits that harmful, like, think of it like an accelerant, right, in a fire. That's the analogy does that work, John?
Yep, that works.
Okay. Think of 15-lipoxygenase acting as an accelerant and Vatiquinone kind of coming in as the fire extinguisher and dampening down this vicious cycle that is further damaging the mitochondria. Vatiquinone's been studied, like I said before, in FA and in also other mitochondrial diseases, and has a lot of safety data and really long-term safety data, more than 10 years of use. So about a year ago, we completed a clinical trial called MOVE-FA. This was a global study to be a registration quality study and that means to hopefully eventually support an approval of the drug in pediatric individuals and young adults with FA. This was a placebo-controlled trial. So when people came into the study, half got Vatiquinone, half got placebo, went on for 18 months, again, enrolled individuals ages 7 to 21. And then there was an additional group of younger children under the age of 7 who were also enrolled in sort of a parallel arm to the study. At the end of 72 weeks, individuals were given the choice to go into an open label extension where additional data would be collected over a longer period of time. And that open label extension has been ongoing. The results of that MOVE-FA pivotal trial. This is a complicated slide. I know there's a lot going on here, but I think if you look at the left-hand side of the slide, what we're trying to show you here, and I'm sure in the back of the room it's hard to see, the primary endpoints the key endpoints that were collected. So the modified Friedreich's ataxia rating scale, which is our standard assessment for neurologic function in the clinic. That modified Friedreich ataxia rating scale, if you've done it, you know it measures lots of different things, and I'll talk about that in a minute, but one of which is upright stability. And then we collect patient-reported outcomes, so your impression of how you're performing on activities of daily living, how fatigue is impacting your life, and then also timed walk tests. What you see here is that the treatment group all did better than the placebo group. And on a few of these outcomes, we reached statistical significance. So more than chance that people improved on these outcomes. And that's statistical significance is important, especially when we start thinking about, regulatory approvals, right? We have to show that this happened because of a treatment effect, not just by chance. When you look at that mFARS, okay, the primary outcome measure, in green here, you see the treatment group, and in gray, it's the placebo group. And what we're showing here is that the people in the placebo group progressed on their mFARS more than people in the treatment group. There's another way to look at this. And like I said, mFARS measures different things. If you've done it, you've had your speech assessed. What is the speech assessment of the mFARS? What's the sentence? Yes, thank you. The president lives in the White House, and well done. You also get to do a lot of fun upper limb assessments, right? Having to point to your nose and the doctor's finger, and then do things like this and things like this. So we assess upper limb function, lower limb function, heel shin slides, and then standing and walking assessments. What we're showing here is that in the study population, the speech assessments, the upper limb assessments, and the lower limb assessments didn't change in the placebo group or the treatment group, right? In young kids, these are not the things that are changing the most. What changed in both the placebo group and the treatment group was upright stability. So standing and walking and sitting. And what you see is that the treatment group changed less or progressed less than the placebo group. John, you want to add additional interpretation to that, or kind of talk to the clinical significance of what we've learned from this study?
Sure. So I'm going to start off in saying that a lot of what we've been able to accomplish has been based on the natural history that you all have taken part of. It provides so much rich information that we could take a look at these subscales and understand how they're driving disease at different parts, at different ages, and what's really assessing how the patients are changing. When we looked at this, and as Jen, I think I'm going to have you present everything because you do such a great job. What we saw was changing was really upright stability. So the only thing we could impact, we did impact. And we've been able to have a conversation with the FDA about this. And I think, again, I'm going to call out Ron, does a fantastic job of getting in front of the people in the FDA and on the Hill and make sure they understand how these scales measure what's important to patients with FA. They were ready for this conversation. They understood the value of upright stability. And when we brought this to them, they were very receptive. We've been able to have a conversation with them. And Jen mentioned our long-term extension study. They looking at this upright stability benefit in MOVE-FA, in addition to the visual acuity study that was done, I think it's 14 years ago at this point. So I think 2010. And then the additional data that we have in the extension that went beyond MOVE-FA. So the MOVE-FA extension study, the FDA is looking at this data, and they're willing for us to bring that forward as a unified package to show that we are impacting all aspects of disease not just in children, which was the primary population here, but also in adults. And we can show that not when we are looking at different aspects of disease, we do then drive change in things like upper limb and lower limb. So our plan is to submit to the FDA by the end of the year, and that is still the plan. And hopefully you guys will see some good news soon.
Yeah. This is the first time we've seen in a clinical trial as well an improvement in fatigue. And that, that was something that jumped out to me the first time I saw the data, because we've been collecting fatigue from the community, fatigue quality of life scales for a very long time. I remember early in my days in the clinic with Dr. Lynch, he always asked that question: if I could fix one symptom, what would it be? And the number of times people said fatigue was initially, when I first started, really surprising. I know it's not surprising to us now, but because we recognize fatigue more as an aspect of the disease, but this is the first time we've been able to see an improvement in fatigue when we've done a clinical study. So I guess the other aspects that are also important are around sort of the overall safety, right, of vatiquinone. And so, John, can you just summarize and speak to that briefly?
Yeah, we have as you pointed out, we've been studying vatiquinone for a long time now, and we have extensive evidence in patients as under 1 out to 68. We've treated them in multiple different diseases, FA and different types of mitochondrial disease, and we can see that we see similar issues between both placebo and the vatiquinone treated groups. And to this point, it's very well tolerated, and we don't see any significant safety signals. So that, in addition to what we've been able to put together for the efficacy, is part of what is really helping us move the program forward.
And maybe the last set of questions around the open label extension that's been ongoing for a while now. Will we learn additional information kind of about the data that was collected in the open label extension? And, will that be part of, future understandings?
Sure. Yeah. When we, and it's kind of, I'll touch on two things here. One, when we first talked to the FDA, we asked them what they needed to see in order to feel comfortable with this data set. And they asked us to ensure that we had about 12 months worth of data in the open label. That essentially means that for the people who started on, for the patients that started on treatment in MOVE-FA, they'd have 3 years of total treatment. And for the patients that were on placebo, they'd have 1.5 years, which is equivalent to what was in our placebo-controlled trial. First off, we have finally completed collecting that data. We're taking a look at it, and watch this space. We'll pretty soon be able to tell you what we saw. The other aspect of that is we were asked, and have been asked in panels like this multiple times, whether or not we would allow OMAV on top of patients within this trial. And because we had this agreement with the FDA to make sure we had that set of data with just monotherapy, after 12 months, any patients in the extension study can add OMAV on top of us and stay within our study.
Thanks, John. All right, I'm going to move on next to talk about Elamipretide. And so, Jean, I've got a little bit of background about Stealth BioTherapeutics in case folks don't know about Stealth. Elamipretide is being developed for several different mitochondrial diseases: Barth syndrome, mitochondrial myopathy, ALS, Duchenne, FA, and there's been more trials, more outcomes in some of those other mitochondrial diseases that we actually learned from and kind of helped inspire thinking about maybe trying Elamipretide for FA. And just a little background about what does Elamipretide do. So again, back to the mitochondria. When there's oxidative stress and the mitochondria are not functioning well, not producing enough energy, it's actually the wall of the mitochondria that starts to take on damage. And the wall of the mitochondria is really important because that's where a lot of the action actually is in terms of the mitochondria function. And so Elamipretide is helping support cardiolipin, which is like an essential part of that membrane. And so when there's oxidative stress, cardiolipin starts to get attacked, the walls of the mitochondria start to get attacked, and Elamipretide is really helping stabilize that cardiolipin and help stabilize that mitochondrial wall. Anything you want to add to that, Jean?
No, just the structure and function is so important. And by being able to restore the structure, you see the improvements in function. So instead of seeing increases, decreases in ATP and increases in reactive oxygen species, you start to see physiologic levels of reactive oxygen species and increases in ATP back towards normal levels.
And so some of the other diseases where you've looked at Elamipretide, I know one of them is a rare disease called Barth syndrome. And in Barth syndrome, there's some overlap with FA in terms of some of the clinical features, specifically cardiomyopathy, as well as some muscle weakness. And in the early Barth syndrome trials, you saw improvements in the cardiomyopathy.
Yes, we saw improvements in cardiomyopathy, improvements in fatigue were one of the things we saw, but it's difficult to measure. But also things like sway and balance and skeletal muscle strength. So, we've been targeting areas where there's a lot of energy consumption, where there's a real need for mitochondria. So things like skeletal muscle, the heart, and with our pipeline compounds, hoping to do more with the brain and neurology.
And so based on some of those results from some of the other studies that you were done, Dr. Lynch went to Stealth and asked if he could do what's called an investigator-initiated study, looking at Elamipretide and seeing if it can help some specific symptoms of FA. And so this study was designed to look at folks who have significant cardiac disease and or vision loss, and see if Elamipretide could help some of those specific symptoms. And so this was a study that, again, Dr. Lynch developed. 16 people were enrolled into the trial. He's got a few other folks, I believe, also on compassionate use, where he's following them to look and see if the drug is going to be helpful in treating some of those symptoms. The study has been completed as of late summer, and I know the team is actively analyzing those results. And so we hope to be able to share those results back with the community soon. We don't have them just yet, but very soon, where I know Dr. Lynch and I email the statistician pretty regularly. Christian, if you're listening, go back to work. So the last thing, Jean, I wanted to ask you about was sort of the status with developing Elamipretide for some of these other therapies. And I know specifically the Barth syndrome, Stealth has submitted that new drug application, right, to request
Yes.
approval for Elamipretide and Barth syndrome.
So Barth syndrome is an ultra orphan disease. The estimated number is about 130 patients in this country that are known to the Barth Syndrome Foundation. So it's a very difficult disease to study to get the kind of statistical power that the FDA would like to see. We've had an ongoing dialogue with the FDA for close to 10 years to try to figure out a way to get this approved. And it doesn't fit into the traditional pathways. And, this week there's an important milestone meeting with the FDA where they bring in independent experts to talk about should this drug be approved? What are the risks? What are the potential benefits? And this is an area where the community, the patient advocacy community, patients who have experience with the drug, physicians, investigators who have experience with the drug have all weighed in. And if there's any chance of approval, it's going to be because of advocacy. So again, it's an important week for us, and it's something people can watch online. If you have any interest, they'll be streaming it live.
Yeah, and these advisory committees have open sessions. And at those open sessions, community members provide direct feedback to the committee and to the FDA about their experience with the condition or with the treatment. And so this really is the opportunity for the patient voice to really be important in the drug review and approval process. We wish you guys the best on the 10th. We're going to be watching. And FARA did submit a letter of support for the Barth syndrome community. Earlier this week, we submitted some written statements and testaments to that. I'm glad for that opportunity. So the last treatment candidate on the pipeline that I want to provide an update on, and you heard a little bit about this in the earlier session, is studying NAD and exercise. And because of those frequently asked questions that Dr. McCormick and Dr. Lynn get about, the benefits of some of these supplements and and exercise, they designed a really amazing study to look at both supplementation of NAD precursors. The one that they're specifically using in this study is called nicotinamide riboside, the dietary supplement that is an NAD precursor. Again, trying to deliver more energy to the cell and comparing that to exercise, because some of these pathways that we're activating with something like NAD precursors, you might be able to activate with exercise. Do I get that right, Shana? Yeah. And so in this study, people are randomized to nicotinamide riboside or exercise or both supplement and, supplement and exercise or no intervention at all, and studied for 12 weeks. And the study enrolled individuals who were 12 to 40 years of age and able to do exercise on a recumbent trike. You can do that in your home on a trainer. The individuals who participated in the study got an unusual study bonus of being able to keep that recumbent exercise bike, which is really amazing. And for people who were randomized to no exercise, were also given the opportunity at the end of the study to receive the exercise protocol, to continue to be monitored, and also have access to a recumbent trike, which is really pretty phenomenal. This study was funded by the National Institutes of Health, right, Jana? And so where are we currently? Shana, you want to kind of give us where we are now?
Yeah, thank you. So I really appreciate that introduction, and one just minor update that we're down to age 10 actually for this study. Yes. I know maybe some younger ones in the audience like, wait a second, I was in this. So yeah, this is my opportunity when I spoke with Jen to really thank the community for so many different reasons. But the first reason is for the idea to pursue this study. We got so many great questions about exercise. I'll quote Kyle Bryant, who's a collaborator and helped us design the exercise intervention. He said, Shana, exercise has got to be good. Like, are you really not going to find that exercise is good? And as a clinician and personally, I have this intuition, exercise must be good, right? But the details of how to do it and how to do it safely and effectively and to individualize it, that is not trivial. That's why we have so many great questions from the community. So the community came up with the idea. And then when we submitted the grant to the National Institutes of Health, which is your taxpayer dollars, which is another really important source of potential funding for Friedreich's research. When we submitted that grant, myself, along with Dr. Lynn and Dr. Lynch, we had support from FARA, and we had natural history data, and we had information about impaired fitness from the community. And that is what helped this project get funded. So we had all that. And then we had advocacy from you all to participate, which has been really, really amazing. We've had so many active and engaged families and participants. So our team, we were required to submit to the NIH several times a year, and I'll recognize they're sitting over there. Anna Didio and Karen McSweeney, who are our lead coordinators on this project, who are amazing. Anna's required to submit to NIH our progress on recruitment. And so on the x-axis here, you see time, and on the y-axis, how many people we've recruited. And we call this climbing the mountain of recruitment, right? Like and I remember the first, we were supposed to begin during the pandemic. So we had a brief delay before we got started. So As of now, as for a goal of 72 folks enrolled with 64 with complete data. So our statistician is satisfied. We've enrolled 61, randomized 61. And so we're really almost there. We've identified all of our participants so that enrollment is closed, and we envision by the end of this calendar year, everyone will have been enrolled so that by early part of next year, we'll be finished and be able to be analyzing the data. So really so many thanks to you all. I think we're going to be able to learn a lot that we thought we would learn about this study, about exercise and about NAD precursors. And then we've had so many more opportunities to learn that we didn't expect over the course of the study, including, for example, we're enrolling people who are on OMAV. Like, how does OMAV work with exercise training? Like, I'm really excited to be able to examine that question, for example. So really, really grateful to everyone for this.
Thanks, Shana, and we're excited for those results. Maybe just one additional question for you. As we're waiting for this data, FARA was also funding a study with a collaborator of yours, Joe Bauer at University of Pennsylvania, and he took a look at NAD precursor supplementation in the FA mouse model. And those results were recently published. Can you just, for the audience, kind of give us a high level, what were the take-home findings from that preclinical animal study?
Yeah, for sure. So one privilege, and I'll represent Joe Bauer and Caroline Perry from Penn, them thanking FARA and the community for support for preclinical research, is as a clinical researcher being able to partner with our basic science colleagues and learning from the animal models and figuring out in an iterative way, how can we apply those insights? It's been really, really valuable. So one of the reasons that we undertook this NAD precursor exercise study was that we found in animal models of Friedreich's exercise is helpful. And in one animal model, the Hershey group at Duke, an NAD precursor rescued, meaning like made better, the heart problems in one animal model. So that's always really intriguing when you have this outcome. And then you go back and you say, well, why did that happen? Like, that's really an interesting problem to have. So we thought, perhaps rightly, that there might be deficiency, meaning not enough NAD in people with Friedreich ataxia. NAD is important for making ATP, so it's plausible that there's not enough. It's also used up in a lot of really important reactions, including antioxidant defenses. So it really could make sense that there might not be enough NAD. But the animal studies have been discrepant. Like, some studies say maybe there's deficiencies of NAD, but other studies say no. In people, it's really difficult to assess accurately. So the study with Joe's group did a different animal model study, again, showed that an NAD precursor was able to help with survival and help with cardiac benefits. Didn't seem to show, though, that increasing the level of NAD was the main reason that happened. Instead, it seemed that the NAD precursor was able to help restore levels of glutathione, which is essential for the body's antioxidant response system. So NAD subserves the ability to kind of restore one of the body's natural and antioxidant systems. So it seemed from Caroline and Joe's data that may be one way in which the NAD precursor is working to help rescue the animal. So then it gives us some ideas of other testing we can do in our human samples to see if that same biology could be operating in people.
Thank you. I'll open it up to our audience. Do you have questions for our panelists? There's one in the back. Oh, Chandra.
Yes. Have you guys considered different types of exercises and how it affects the mitochondrial, energy? Because it seemed like there was the trike was the primary one in that example.
Yeah, I'm really appreciative of your question. So the question, have we considered different types of exercise? Because there's lots of different ones. So the trike is the focus. There's 3 sessions a week of an individualized cardio protocol. So we have people do an exercise assessment in the lab, so we can give them a target intensity heart rate wise to complete those sessions. We also having folks complete 2 strength sessions a week, really because as clinicians, our intuition was probably that a combined form of exercise would be most appropriate. And our public health guidelines say that we should be using both. But to the questioner's point, like it may be that strength should be our focus, especially if we're thinking about, for example, preventing falls and promoting bone health. So this is my plea to the group. Our team is sitting there. I'll be here. NIH has asked us about what we want to do next. And one question I have is about strength training, because a lot of our participants seem to really enjoy that. And a lot of members of the community seem to like that. So we chose a combined approach because we thought that was probably the best one, but unpacking different types is absolutely a great question for future.
Chandra.
So my question is for each of the panelists. Are there any preclinical studies on the effect of the combination of OMAV in your product or supplement? And if there are, can you briefly describe them? Like what they showed?
We're really kind of focused on clinical now. I think the first evidence we'll see would be from the combination that we're seeing in the open label studies. I think we do look in some preclinical data to see how these two compounds can interact. They do work through similar pathways, although not identical. The 15-LO and how that impacts lipid peroxides and can reduce depletion of glutathione, I think, is on one side, and then the ability for NRF2 inhibitors to upregulate glutathione and overall drive forward, improve oxidative stress responses. I think there are parallels there. We don't quite have evidence we can share right now or data we can share at this point, but it is something we are looking at. So
Thank you.
I'm happy to add to that. From the USPI, or the approved label that we have for Skyclarys Omaveloxolone, we do know from the earlier research that there are drug-drug interactions, particularly through the CYP450 pathway. So there are specific drugs that we would know we'd want to avoid. But when we consider not just the potential mechanism of action, but how, going back to the pharmacokinetic piece and how those drugs are metabolized in your system, they might actually interact and change the levels of the different drugs in your system as well. So that's something that with any therapy, when you're starting or maintaining, if you're adding different supplements or vitamins or other potential investigational or approved therapy, that's a conversation to always bring forward to your healthcare provider to make sure that they can evaluate that for the potential drug-drug interaction as well.
Yeah, and maybe I'll just also add to this. FARA has funded, I think, 3 or 4 different research projects this year, looking at combination therapies in both the cell models, so the iPS-derived neurons and cardiomyocytes, as well as in the mouse models. And they're combinations of some of the therapies that we just talked about in the panel, but also others as well. And so hopefully several months from now, we'll be able to start sharing some of those results. But lots of activity, lots of investigators asking Chandra that exact question. And, we have the model systems, I think, in place to start doing some of that. And that's been a priority for us. Well, I just want to thank all of you for participating today, for all of the work that you do to continue to bring treatments forward for FA. It's exciting to hear that we're also going to have a second drug being submitted to a regulatory agency for approval before the end of the year. Wow. So thank you all. And if you still have questions for our panelists, they're going to be here. Their colleagues are here all day. So please, during the break, take an opportunity to talk to them and ask them your questions.